Formally start glycolysis. One step, just conversion of glucose to pyruvate, but that's not simple. There are 15 reactions in it. There are 15 reactions in it. 15 reactions in it. And we will study each of that reaction in detail. And each of reaction has its significance. And don't worry. You will learn it once you finish listening to this video. Alright? Come back to it. So first step in the glycolysis is the entry of glucose into the cells through blood. The first step is the entry of glucose into the cells. So this is the entry. Glucose enters into the cell by crossing the blood, and that glucose is there. But glucose is a very nutty boy. This is really a very nutty boy. And if we don't trap it, it will move out. So the second step, the first step of glycolysis is entry, and the second step is trapping. If we don't trap this naughty boy, it will leak out of the cell, and that glucose is trapped by how? That it is phosphorylated. It is phosphorylated to make glucose 6-phosphate. It is phosphorylated, and whenever the word phosphorylation comes, the enzyme will always be kinase. So here the enzyme here is hexokinase or glucokinase. What are their stories? I will tell you in a while. Hexokinase or glucokinase. But in this process, that phosphate group uses ATP and ADP. Actually, there were 3 phosphates and now only 2 phosphates. ADP, 1 phosphate, is attached to the glucose. In this way, the glucose is trapped. And important, this is irreversible step. I'm placing a star in here. This is irreversible steps. Once glucose 6-phosphate is found, glucose will not be formed back, and it will be trapped. So the phosphorylation of glucose, in fact, traps glucose. And which enzyme is responsible for trapping? Glucokinase or hexokinase. Glucokinase is present in liver, while hexokinase is present all where. Right? The first step. This is the first step. Now the second step. The glucose 6-phosphate. Glucose 6-phosphate is converted to fructose 6-phosphate. Alright? Glucose is aldose sugar and fructose is ketose sugar. Both are hexoses. And the enzyme here is just isomerase. And this is reversible step. Okay? And the third step, this fructose 6-phosphate. This was the third step. The formation of fructose 6-phosphate, and then this fructose 6-phosphate is converted into fructose 1,6-bisphosphate. 1,6-bisphosphate. Alright? And once this happened, so there was just one phosphate group, but now there are 2 phosphate groups. Why he used this bis? He may have used di, but bis means bi means 2, while S means separated. Right? So fructose 6-phosphate is converted into fructose 1,6-bisphosphate. Again, a phosphate group is added. Actually, it is phosphorylated, and once one thing is phosphorylated, which enzyme is there? Kinase. Which enzyme is there? Kinase. And the enzyme is called phosphofructokinase. It is also called PFK. It's not JFK. It's PFK. You know JFK? It's an airport in America. Yeah. But here this is PFK. Alright, phosphofructokinase. And there are 2 types of phosphofructokinase. And here, which phosphofructokinase is present? The phosphofructose kinase 1. Alright? Phosphofructokinase 1. And this is the second step which is irreversible, which is irreversible. This is important because this is important to understand gluconeogenesis. Okay, so you have to understand the first step, which is irreversible, is the formation of glucose 6-phosphate from glucose, while the second step, which is irreversible, is the formation of fructose 1,6-bisphosphate from fructose 6-phosphate. Okay, I hope this is clear. Now, the fourth step. The fourth step is that fructose, there is just one periphery of this thing, and that is important, and that what is the role of PFK2, phosphofructokinase 2. I explain in a while, but before going into that, let we see what is going to happen with this fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. This fructose 1,6-bisphosphate is cleaved, is broken down. It has 6 carbons. Oh, I forgot to mention one thing, that if this is the phosphorylation, then another ATP must be used here, and that ATP will be converted into ADP, and this is the second irreversible step, and it requires ATP. Second irreversible step, and it requires ATP. All the MCQs, all the questions related to glycolysis will be asked from these things. You know, what are the irreversible steps of the glycolysis? The one, formation of glucose to glucose 6-phosphate, conversion of glucose to glucose 6-phosphate. The second, conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. And both require enzyme. Which enzyme? Glucokinase, hexokinase in the first, and in the second, PFK1, phosphofructokinase 1. You see that now? Fructose 1,6-bisphosphate. You see here, fructose 1,6-bisphosphate, 6 carbon. Fructose 1,6-bisphosphate, 6 carbon. They will be broken down into 2 3-carbon compounds. One will be aldo sugar and one will be keto sugar. And 3-carbon sugar is triose. And if it is aldose, it will become aldotriose. And that aldotriose is called glyceraldehyde. And the other will be ketotriose, and that ketotriose is called dihydroxyacetone. Alright, you see that? Now the fructose 1,6-bisphosphate is broken down to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And in biochemistry we are fond of making words, and so this is also called DHAP. What? DHAP, right? And we say this G3P. Okay, and the enzyme, and see that I have placed arrows on both sides. It means this is reversible. And the enzyme is called aldolase.